Hello everyone, I'm Fatma Najib, and me and my colleagues Selma Jabnoon and Miriam Rosenbourg are going to talk about our research, which is bioassay guided fractionation and cytotoxic activity of Borago officinalis flower, under our supervisor Dr. Adil Farouk Wali in the Department of Pharmaceutical Chemistry in Iraq Medical and Health Sciences University in Ras Al Khaimah. All around the world, cancer is a major cause of death, representing 13% of all deaths around the world in 2018. And breast cancer is the second most common cancer diagnosed among women in the world, as World Health Organization indicated that there may be 21.4 million instances of cancer and 13.2 million deaths due to cancer every year by 2030. There is an urge for the expansion of a new anti-cancer medicine for its treatment and prevention. Medicinal plants are the fundamental sources of the neutral bioactive compounds. 50 to 60% of anti-cancer compounds are derived from medicinal plants. There are still many of plants that have anti-cancer potential, but they have not yet been fully investigated. And the current investigation carried out a screening of extract and various fractions of Borago officinalis flowers were investigated for antioxidant and anti-cancer activity against breast cancer cell lines MCF7. Our plant name for this research is Borago officinalis. It is from Boragoniaceae family. The part we use for this research is flower. Traditionally, Borago is used in the treatment of various diseases such as multiple sclerosis, diabetes, heart diseases, and eczema. The plant contains a variety of bioactive chemical constituents, more than 150 volatile and aroma-yielding compounds, like glycosamine, gamma-linolenic acid, and quercetin.
For our aims and objectives, we have identified, authenticated, and extracted Borago officinalis flower. We did the phytochemical investigation of Borago officinalis flower using liquid chromatography mass spectroscopy technique. We have collected different fractions from the extracts using fractionation column chromatography techniques. We did in vitro antioxidant screening of the extracts and fractions. And then we did in vitro assay for the cytotoxicity using MTT assay for MCF7 cancer cell lines. Now for our methodology, the Borago officinalis flower was collected from Shiraz, Iran. The dried flowers of Borago officinalis was extracted by sonication extraction technique, which was kept for the temperature of 40 degrees Celsius for 35 minutes under the power of 60% using 100% methanol. The extract obtained was concentrated to dryness using Rotra evaporator. The concentration of phenolic contents for the extracts and fractions was determined using FC spectrophotometric method, whereas for the total flavonoid content, it was determined by aluminum chloride colorimetric method. The free radical scavenging activities of the extracts and fractions were evaluated by DPPH assay and ABTS assay. The cytotoxicity activity was performed as described earlier by Yusra et al.
And now for our results. We fractioned our methanolic extract using column chromatography, where we obtained three different fractions: the butanol fraction, the aqueous fraction, and the ethaloacetate fraction. We determined the total phenolic and flavonoid content of our extract and fractions. The total phenolic content was determined in milligram gallic acid equivalents per gram of the extract, and the total flavonoid content was determined in microgram quercetin equivalents per gram of extract. For our total phenolic content, the highest concentration was found in the aqueous fraction with 321, followed by the butanol fraction with 300, and then the ethaloacetate fraction with 254. And finally, the least phenolic concentration was found in the methanolic extract with 218. As for the total flavonoid content, the highest flavonoidal content was found in the methanolic extract with 165, followed by the aqueous fraction with 123, and then the butanol extract with 115, and finally the phthaloacetate fraction with 86. We determined the in vitro antioxidant activity of our extract and fraction using two methods, the DPPH assay and the ABTS assay. As you can see in our tables, the aqueous fraction had the highest radical scavenging activities in both assays, followed by the butanol fraction, then the phthaloacetate fraction, and the least radical scavenging activity was observed in the methanolic extract in comparison to our standard, which is the butylated hydroxyanisole. We conducted liquid chromatography mass spectroscopy in our methanolic extract for phytochemical profiling, where we found a total of 5 bioactive molecules that we analyzed to correlate molecular ions with fragmentation patterns. We compared our data to reference compounds mentioned in literature databases and computer repositories. As shown in our figure, we found terpenoids, carotenoids, polyphenolic, and fatty acids compound among our bioactive molecules. We conducted an MTT assay on cancer cell lines MCF7 to find the half maximal inhibitory concentration of our fractions and extracts. This test is conducted to find the antiproliferative activity on human cell lines. This test is essential for safe treatment. It allows us to identify the intrinsic toxicity of the plant and the effects of acute overdose. We used 5-fluorouracil as our standard, which gave a value of 9.23. As you can see in our table, all of our extracts and fractions showed cytotoxic activity. However, the highest cytotoxic activity was observed in the aqueous fraction with 12.01, followed by the butanol fraction with 17.73, and then the methanolic extract with 23.91 micrograms per ml, and finally, the least cytotoxic activity was observed in the phthaloacetate fraction. These images present the MCF7 cancer cell lines after 48 hours of incubation with our aqueous fraction extracts. This shows the antiproliferative activity obtained in the aqueous fraction, which indicates that the Borago officinalis shows anti-cancer activity. The data displayed is the mean of the test as measured in triplicate. There is no such critical alterations like cell shrinkage and reduction in cell population seen in the aqueous fraction when treated on the normal cells, which indicates that the non-toxic nature of the compound towards the normal cell lines. We have also monitored cell morphological changes that might be associated with the methanolic extract treatment. The study reflected that the MCF7 cancer cell lines, when observed under light microscope, show a decrease in the cell count, which increase in the concentration of the aqueous fraction. In conclusion, our findings suggest that the Borago officinalis extract possesses potent antioxidant and cytotoxic activity and anti-cancer properties, which is possibly due to the principal bioactive phytochemical composites existing in this plant. However, additional studies are needed to identify how these natural extracts can be used as complementary approach to currently used chemotherapies of different cancers. Also, more study designs to investigate the molecular mechanism underlying anti-cancer activity are needed. Our team is working on the isolation of bioactive molecules in Borago officinalis, which is responsible for the cytotoxicity. They can be considered as good substrates for future structure-activity relationship studies and modifications to produce more potent cytotoxic derivatives. We would like to thank Dr. Rao, the University President, Dr. Padma, Dean Rakops, and Dr. Adil Farouk, our supervisor, for the kind support and encouragement. These are our references, and thank you.